Welcome to Task 4 of the subject Biology of Engineers on Hydrogen Bonding as the Basis of Life. Introduction. Hydrogen bonding is a weak electrostatic attraction which arises when a hydrogen atom is surrounded by atoms having high electronegativity such as fluorine, nitrogen and oxygen. The two atoms are bonded by covalent bond in which higher electronegative element attracts more number of electrons, making it negatively charged, and hydrogen is positively charged. Hydrogen bonding can take place in same molecule or between two different molecules. It is stronger than van der Waals interaction and weaker than covalent or ionic bonds. Hydrogen bonding in water. Water exhibits cohesion due to hydrogen bonding. It is useful in plants as it helps in transporting of water and other nutrients up the plant. Hydrogen bonding is responsible for surface tension in water. It enables the surface of water to withstand pressure. Hydrogen bonding gives water viscosity. It allows water to dissolve with other solvents, thus making it soluble. Water has high specific heat due to hydrogen bonding. It leads to absorbing of heat when water molecules break and release of heat when they form, minimizing temperature change. Number of hydrogen bonds needs to be broken to evaporate water, giving it high heat of vaporization. Hydrogen bonding in water. Hydrogen bonding in DNA. Hydrogen bonds helps in holding two strands of the DNA double helix structure together. DNA contains four bases: guanine, cytosine, adenine and thymine. The reciprocal base sets of guanine with cytosine and adenine with thymine are connected to each other using hydrogen bonds. This hydrogen bond holds the two strands of DNA together. Millions of such bonds makes DNA stable molecule. Two strands of DNA fit in with a helical structure due to hydrogen bonding on phosphate branches of each nucleotide. It introduces hydrophobic effects that stabilizes DNA by keeping the hydrophobic bases inside the helix and polar outside where it is in contact with water. 3 hydrogen bonds in guanine and cytosine. 2 hydrogen bonds in adenine and thymine. Hydrogen bonding in proteins. Hydrogen bonds helps in holding polypeptide structures together in alpha helix and beta conformation secondary structures. Secondary structures in the hydrophobic core of protein has hydrogen bonding between main carbonyl oxygen chain and amide nitrogen, which gives directional interactions that support protein collapsing, protein structure and subatomic acknowledgement. Helical structures of polypeptide chain contains residues that are stereochemically proportional. The interatomic separations and bond edges of values found in amino acids, peptides, and other basic substances identified with proteins, and the conjugated amide framework is planar. In structure with 3.7 residues per turn, every carbonyl and amino group is joined by a hydrogen bond to reciprocal group in third amide group in polypeptide chain. Plan of 3.7 residue helix. Plan of 5.1 residue helix. Hydrogen bonding in antibodies. Hydrogen bonding helps antibodies in synthesis and discharging by B cells to tie to their antigens such as proteins, DNA and connection happens by non-covalent forces between the antigen consolidating site on the acting agent and epitope. These are imperative in human body as it encompasses pneumococci and shields it from phagocytosis. Photomicrographs. Hydrogen bonding in enzymes. Hydrogen bonding helps enzymes to bind to their antigen. Presence of enzymes is essential in human body. They perform controlled reactions with food as it enters our mouth, which are required for maintaining life. Their entire reaction relies upon perceiving and clinging to particular reactants, which happens with the assistance of hydrogen holding. Complementary dipoles on the surface of enzyme precisely coordinates with that of reactant, which bond to particular reactant because of specific arrangement of dipoles. Hydrogen bonding in transcription factors. Hydrogen bonding helps transcription factors to bind to each other and with DNA. Translation factor is a protein which cooperates with restricting destinations utilizing a one-of-a-kind blend that is fundamental in human body as it controls the rate of interpretation of hereditary data from DNA to a wrong person by authoritative to a specific DNA succession. Conclusion. Hydrogen bonding is weak forces compared to other known forces, but it plays a very important role in human body. It is a minimum requirement for human existence. It is everywhere, be it enzymes, antibodies, DNA and proteins. It plays a major role in providing human body energy, transferring DNA further, shield pneumococci and maintaining double helix structure of DNA. Hydrogen bonding is a basis of life and its presence is mandatory. Thank you.